Hi, welcome to Biology classes with Lakshmi Chandra. In today's session, we'll learn about a new concept. We'll learn about recombinant DNA, how it is produced, and its importance in biotechnology. So let me just take you first regarding the meaning of a recombinant DNA. In the word itself, it is very much clear. Recombinant. That means the combination of DNA is changed. There is some change in the combination of DNA. And that is what is needed in biotechnology also, in molecular biology part. Imagine you have a usual DNA. I'm just drawing a segment. Okay. Into this, if I am introducing another DNA molecule, say this is another DNA molecule, okay, I am taking a segment of this DNA and I am inserting this with the original DNA here. So it will be something like this. A segment is taken from here and it is put in the place here. It is inserted here. Okay, this segment I have transferred to this DNA. Now I'll call this DNA as a recombinant DNA because its combination is changed. Now why I have inserted this segment here is because this segment is having some importance, some features which I want to be with the original DNA. I'll just give you one small example. Imagine I want to produce insulin, the hormone insulin, inside a bacterial cell. So what can I do? I'll search for the DNA segment which is producing human insulin. That DNA segment I'll take and I'll insert it into the bacterial chromosome. So what that chromosome of bacteria has become now? It has become a recombinant chromosome because I have inserted a gene, that is insulin producing gene or insulin producing segment of DNA into the bacterial chromosome. So now the bacterial chromosome is a recombinant molecule. I hope you understood the concept. So whenever we change the combination of a molecule, you call that molecule as a recombinant molecule. So in this case the molecule is DNA, so I can call it as a recombinant DNA. If it is a protein, I'll call it as a recombinant protein. RNA as a recombinant RNA. Likewise. So whenever a foreign gene, so this gene is a foreign gene here. This is foreign. This gene is introduced into another one. So for this DNA, the gene which I have drawn here in blue color is a foreign gene, or this foreign gene is our gene of interest. This is the gene which we want to be here. This is our gene of interest. So in the example I gave you is insulin producing gene is my gene of interest, or that is a foreign gene for the bacterial cell. So what I did here is I have inserted this foreign gene, that is insulin producing gene, or another way I am calling it as gene of interest, into the chromosome of bacterium, so that bacterial chromosome have become a recombinant molecule. Same way, if I am introducing this foreign gene or gene of interest into a cell, I'll call the cell as transformed cell. If I am inserting it into a molecule, I call it as a recombinant molecule. But if a cell is carrying a foreign gene or a gene of interest, I'll call that cell as a transformed cell. So that bacterial cell has become a transformed cell now. Its chromosome is recombinant chromosome, and a bacterium carrying a recombinant chromosome, I use the term as a transformed cell. Now, if I am inserting this foreign gene or gene of interest into an organism, say a plant or an animal, into a whole organism's body, it is there, I can call that organism carrying a foreign gene as a transgenic organism. So here, as you can see, the terms are only different. If it is with respect to a molecule, I call it as a recombinant molecule. If it is with respect to a cell, I use the word transformed. If it is with an organism, I use the word transgenic. Meaning is same. So to produce a recombinant DNA, what are the steps that we are going to follow? That is the essence of today's session. Let's see the steps one after the other. Steps for the production of recombinant DNA. So here you will come across some new terms. Don't worry, all those things will be explained to you in detail. First step, isolate the foreign DNA. So now when I use this word foreign DNA, you know this is my gene of interest. So you're isolating it. How to isolate the DNA will be dealt with in a different session. Okay. So first step is you isolate a foreign DNA. So I am drawing here like this. This is, imagine, a foreign DNA. Okay. Now next step, isolate also a vector DNA. Isolate a foreign DNA, I think now you all understood. Now next step is vector DNA. Vectors in biology means carriers. Okay, vectors of diseases we all know. For example, the disease malaria, vector is female Anopheles mosquito. So likewise, vector means carrier. In molecular biology also, vectors means carriers. Carriers of molecules. Okay, carriers of molecules. So here I have isolated a foreign DNA. I am also going to isolate a vector DNA. That means a carrier or a DNA which can carry another DNA. Okay, vector means carrier. Here vector is in which form? It is in the form of a DNA. So I am going to take a DNA molecule which can carry another DNA molecule. That is vector DNA. Okay, so here the vector DNA I have taken the example as plasmid. Plasmid as a vector DNA. So let me just tell you what a plasmid is. This is very important for you in this CBSE examination. Okay, plasmid. Often its definition or its features are asked. So let us see what a plasmid is. Plasmid is usually found inside a bacterial cell or a prokaryotic cell. In few cases it is also found inside eukaryotic cells. Okay. So you know this, I have just drawn a bacterial cell, and bacterial cell is having its own chromosome. Okay, I am just drawing it like this. It is found in a place called as nucleoid. We all know regarding that. Apart from this, so this is its usual chromosome. It is having DNA. You know chromosomes are made up of DNA and proteins. Okay. Apart from this DNA, the bacterial cell also has another DNA which is usually circular and it is double stranded and it is found outside the usual chromosome. Okay, so plasmid, first feature is it is extra chromosomal. Extra chromosomal means it is found outside the chromosome. Extra chromosomal. What is the next feature? It is circular. It is double stranded. Extra chromosomal, circular, double stranded DNA molecule it is. And what is the importance of this plasmid for a bacterial cell? It imparts or it gives some additional properties. It imparts or it gives some additional properties. to this bacterial cell like antibiotic resistance. That means, suppose this bacterial cell is having the plasmid and this plasmid is having a region, this region here. Okay, this region is enabling this bacterial cell to resist the effect of one antibiotics, say common antibiotics. I'll take penicillin. Penicillin is an antibiotics. Okay, that means if you add penicillin to a culture of bacterial cell, the bacteria will die. But this particular bacterial cell will not die. Why? Because it is having a plasmid, and this plasmid is having a region which can resist the effect of penicillin. Even if penicillin is there, nothing will happen to this bacterial cell because this bacteria is able to resist. If you take out this plasmid from here, this bacterial cell will die because that additional property is gone. Okay, so what are plasmids? Very simple. Extra chromosomal, double-stranded circular DNA molecules which imparts some additional properties. So that is a plasmid. And usually in recombinant DNA, we take plasmids as vectors. There are different kinds of vectors, phagemids, different kinds of vectors are there. But for class 12, we are concentrating on plasmids. Okay, more are there to tell you on plasmids regarding a particular kind of artificial plasmid and all. That will be dealt in the coming sessions very soon. So here, our first step, I have isolated a vector DNA, a foreign DNA, and then also I am going to isolate a vector DNA, and here I have taken it as plasmid. So having understood what a plasmid is, now I can draw that also. Okay, so this is foreign DNA. I have also taken a vector DNA. I have taken plasmid. So you know plasmids are double-stranded circular DNA molecules. So I am drawing like this. So this is my first step, isolating the foreign DNA and isolating the plasmid. Okay. Now next step, you can see here, digest the isolated DNAs using the same restriction enzyme to get sticky ends. Now here one has to pay attention. First important thing is you are using the same restriction enzyme. Often it is asked, why we are using the same restriction enzyme to digest the foreign DNA and the vector DNA. Digest means to cut. Okay, to cut the foreign DNA and to cut the vector DNA. Why we are using the same restriction enzyme? Answer is here itself. It is to get sticky ends. Now comes the point. What is the meaning of sticky ends? In order to understand this very clearly, I would suggest you to refer regarding the previous videos regarding restriction enzymes and how they are action of restriction enzymes. Okay, then you will understand clearly what sticky ends are. But for your knowledge, let me just tell you, imagine you are having a DNA with GAATTC as the recognition site. Okay, so this is a palindromic sequence. I hope you remember what a palindrome is. It was all dealt with in the previous videos. Okay, 5 prime 3 prime end I have written here. Now here I am using the restriction enzyme EcoR1. I hope you remember how I have named this as EcoR1. And when this enzyme is acting on this particular sequence, it is making a cut here between G and A. Here also between G and A. So on both the strands, it is making a cut. Okay. Now after the cut is done, let us see how things will be. It is beginning like this. 5 prime G. Then there is a cut. Okay, these two are joined here with hydrogen bonds. Now after G there is a cut. So A is separated, AA, TTC, 3 prime, it is separated. Now this joint, this G is joined with C here, then comes T, T, A. After A there is again a cut, then comes G, 5 prime, likewise. Okay, so here this G is joined and you have things like this. So here, if I just draw it, it is like this. You have here G joined to C down, and then there is a segment like this. And here the G is there down, I am drawing from here, and there is a segment like this. So there is a space here, you can see, there is a space, another one here, that also you can see here. So this is like two L segments, they are joined one after the other like this. Okay, there are overhangs, this is hanging like this is also hanging like this. If you want to join them, they will naturally join by hydrogen bonds. So where is the cut? Cut is here, between the phosphodiester bond. Okay, so when you cut using this restriction enzyme EcoR1, it is going and making a cut here, it is making another cut here. So what you are going to get is you are going to get two ends which are hanging over one another. Such ends are called as sticky ends. Sticky ends. Now I will also give you an example where the sticky end is not coming. Then you will understand better. Okay. I will take another palindromic sequence. 5 prime, G, 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 C, 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 3 prime. Okay. Again 3 prime, C, 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 G, 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 5 prime. So this is a palindromic sequence. Here I am adding a restriction enzyme. I am just putting it as R2. Just a vague name I am giving. Restriction enzyme 2. This restriction enzyme 2 is making a cut between G and C. So in the other strand also it will make a cut between G and C. Now see what kind of ends they are going to make. So here it is 5 prime, G, 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 then there is a break, then comes C, 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 3 prime. Down it is 3 prime, C, 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 then there is a break here, then comes G, 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 5 prime. Okay. Now if you just draw it, they are joined like this. So this is one block, this whole thing. This is yet another block. There are no hanging ends. Earlier we had hanging ends like this. One end is here, another one is going like this, and then there was another end like this coming down. These were hanging. But here you can see there is no hanging ends. But they have to be joined here very solidly. So this end there is no overhangs. There is no overhangs here. So these ends are called as blunt ends. But earlier, as we have seen here, you can see these hanging ends. So there are overhangs. So when a restriction enzyme is coming and cutting and you produce overhangs like this, you call such kind of ends as sticky ends. I hope it is clear to you the meaning of sticky ends and blunt ends. Now coming back to our point, digest the foreign DNA as well as the vector DNA with the same restriction enzyme to get sticky ends. Okay, so sticky ends is preferred in recombinant DNA. The sticky ends is preferred over blunt ends because they can directly join with hydrogen bonds. And then you just use the enzyme ligase here, it will join automatically. You need not use much of a strain to join the two strands. Okay. So let us see. So you have understood the meaning of sticky ends. Now again, why the same restriction enzyme is because, see, you have taken the foreign DNA. This is our foreign DNA. This is our vector DNA. Fine. Now what are you going to do? Next step, digest. Digest the foreign DNA and the vector DNA. Digest means you cut. You're cutting this foreign DNA here and here you have cut. Vector DNA you have cut here. Okay. So how are the things now? Let us see. You have cut the foreign DNA. You got 3 fragments. Vector DNA you have cut here at this end. So it is opened like this. At that end it is opened. Okay. Now what we want? We want one of these segments to be inserted here. Okay, we want one of these segments to be inserted here. For example, imagine if this segment is having that insulin producing gene. Insulin producing gene. I want this gene or this DNA fragment to be here. Okay, this DNA fragment I want to place it here. So first what I should do? I should separate this insulin producing fragment, isn't it? I have taken the foreign gene, I have cut it using restriction enzyme. Now I want my gene of interest. For that, I have to separate the foreign DNA fragment with the gene of interest. I have to separate the foreign DNA fragment which is having my gene of interest. Now how do I separate that? I separate it using a method called as gel electrophoresis. A detailed class regarding that process, that technique will be taken to you very soon. Okay, so just understand gel electrophoresis is a separation technique that is used to separate the DNA fragments based on their size. So based on the size, here also I separate the DNA fragment so that I get the fragment which I need. Okay, so I have isolated the foreign DNA, I have isolated the vector DNA. Now next step, what did I do? I had cut or I digested the foreign DNA and vector DNA. Now I am isolating or I am separating the gene of interest using gel electrophoresis technique. So this is what I need. Among these 3 fragments, I need this fragment. Okay. Now here I have my vector DNA also cut open. Now since I have cut open this vector DNA and the foreign DNA with the same restriction enzyme, what I am going to have is I am going to have a sticky end. Okay, so that is the importance. Usually in recombinant DNA, we cut the foreign DNA as well as the vector DNA with the same restriction enzyme in order to get the sticky ends. So here both of them are having that overhangs. So I can easily insert this particular foreign DNA fragment into my vector DNA. It is inserted here. Foreign DNA is now here in the vector DNA. So now what will I call this vector as? It is having a foreign gene, isn't it? So I will call this vector as a recombinant vector. Now simply I cannot just keep it here. I have to join it. So how do I join? How do I join the foreign DNA with the vector DNA? Using the enzyme ligase. Okay. So I hope you understood the different steps in a recombinant DNA technology. So first step, you isolate the foreign DNA and then you isolate the vector DNA. In this case, the vector taken is plasmid. Okay. Next step, you digest the foreign DNA as well as the vector DNA with the same restriction enzyme so as to get sticky ends. Then you just isolate the gene of interest using gel electrophoresis and then join the gene of interest with the vector DNA so as to get the recombinant vector. Now I told you vector is a carrier. So now this recombinant vector will carry our gene of interest into a cell. So which cell I need it to be in? I want this to be inside a bacterial cell. So now this vector DNA is carrying my gene of interest. Okay, my gene of interest, that is the insulin producing gene, is now inside a bacterial cell. Now what will I call this bacterial cell as? You call this cell as a transformed cell. Transformed cell. Cell carrying a foreign gene. Molecule carrying a foreign gene is called as recombinant. So recombinant vector is now here. Only because it is a plasmid, it can carry it inside the bacterial cell. So I hope you all understood regarding the different steps. In the next session, I will tell you regarding plasmid, what a plasmid is, regarding recombinant plasmid, how it is produced in artificial plasmid, and also regarding gel electrophoresis. So do watch the sessions without fail. Very soon I will be coming. Okay, and I'm very happy with the overwhelming responses that I'm getting. I'm very thankful also for your support. Do subscribe the channel. Pass the message among the needed ones, your friends, so that they also are getting benefited, and keep encouraging like this with your positive responses, and also feel free to ask doubts if any. Thank you all.